So actually these steps, it shows the process of transcription. Okay, so how the transcription is carried out. The first step is initiation, elongation, and termination. So if you take the bacteria or the prokaryotes, so what is happening here? The synthesis of all types of RNA are catalyzed by a single RNA polymerase. So this is another difference between prokaryotes and eukaryotes. Okay, here the synthesis is catalyzed by a single RNA polymerase, and it involves 3 steps: initiation, elongation, and termination. So what is happening in initiation? So this is the RNA polymerase enzyme that binds to the promoter region. Okay, promoter region in the DNA. So here this RNA polymerase, they have different subunits. Actually, the subunit is, that is, we can say it as core enzyme and hollow enzyme. So actually the core, it needs the factor that is called the sigma factor for initiation to happen. Okay, so this enzyme, the whole enzyme is called the hollow enzyme, and the core enzyme plus this sigma subunit is called the core enzyme, is that is without the sigma subunit. Okay, the core enzyme plus sigma subunit is called the hollow enzyme. So this RNA polymerase in prokaryotes, so it consists of many subunits like 2 alpha subunits, beta, beta dash, and omega subunits. They form the core enzyme, and this sigma subunit is needed for initiation to happen. Okay, so this is the RNA polymerase which binds to the template strand. Okay, so it binds to the template strand, that is the process of initiation. So here the RNA polymerase enzyme binds at the promoter site of DNA. Okay, so only at the promoter region the enzyme will bind. So as a result of binding, what is happening? Local unwinding of the DNA double helix. So at that site there occurs an unwinding of DNA helix. So if you see here, you can observe only at that point after the RNA polymerase binds, there occurs an unwinding, that is splitting of the double helix. Okay, so then initiation factor, sigma factor present in RNA polymerase initiates the RNA synthesis. So that enzyme is called the hollow enzyme, that is core enzyme plus sigma factor. So this is the process of initiation. Then the next stage is the elongation. So what is happening in elongation? It is synthesized in the 5' to 3' direction. And in this process, activated ribonucleoside triphosphate, that is ATP, GTP, UTP, and CTP are added. So all the ribonucleoside triphosphates are added, and this is complementary to the base sequence in DNA template. So this is the elongation. So in the elongation, here there occurs a formation of transcription bubble. This is called the transcription bubble. So only 8 to 9 bases will be attached to the DNA here, and the remaining RNA will leave the DNA. See here, after it is synthesized, it leaves the DNA. So this is the RNA polymerase enzyme, and the sigma subunit is released after the initiation is complete. So this is the elongation process where the nucleotide addition is happening with the help of the enzyme RNA polymerase. So this is the elongation. Then next is termination. So how the termination is happening? That involves another factor which is called the Rho factor. Okay, RNA polymerase with Rho factor, it causes the termination, and the synthesized RNA is released. So termination factor or Rho factor, it binds to the RNA polymerase and it terminates the transcription. So these are the different steps which are involved in the transcription. Okay, initiation, elongation, and termination. Okay, clear?